Hello everyone, my name is Brendan Nolan Fisher and I am a current third-year undergraduate student in the Department of Biomedical Engineering. Today I am here to talk about bioprocess optimization for the expansion of equine mesenchymal stromal cells through the elimination of a seed train. To begin, I'd like to start by identifying the problem as well as a possible solution. Currently, at the University of Calgary, there is a collaborative study taking place between the Department of Veterinary Medicine as well as the Department of Biomedical Engineering. Their goal is to create an accurate animal model for cartilage injury and repair. As such, mesenchymal stromal cells, also known as MSCs, have been chosen as a possible therapy source based off their immunomodulatory properties as well as their ability to differentiate into cartilage. One of the main challenges facing this study is the ability to produce clinically relevant cell counts in a short period of time. Typically, this is done via static conditions. However, this is very labor intensive and there's often a high viability between each flask. Our solution to this was stirred suspension bioreactors. These are well-mixed vessels that are very easy to scale and they are much less labor intensive than their static counterparts. As such, we move on to the bioprocess that was designed around these reactors. Currently, cells are thawed from cryogenic preservation into a static dish termed a seed train. This seed train prepares cells to be seeded in a dynamic culture system such as our stirred suspension bioreactors. In our bioprocess, they are serial passage from 100 milliliter vessels into 250 milliliter vessels before being harvested for reinjection, or in the case of my research, being frozen for later use. The goal of my research was to completely remove the seed train from the bioprocess. The removal of the seed train would allow us to create a shorter, more clinically relevant bioprocess that reduces the amount of time from thawing of cells to reinjection and treatment of injury. Based off of this bioprocess, the following experimental outline was created. Cells from two donors were used in this study, one male and one female. Both cell lines were sent through two conditions, a seed train condition, where cells were initially thawed into a seed train before being used to seed a 100 milliliter spinner reactor, followed by a 250 and then harvested for cell validation, and a direct thaw condition, where cells are directly thawed into the 100 milliliter spinner before being passaged into a 250 milliliter spinner, and then again thawed for cell validation. Three methods of cell validation were chosen: trilineage differentiation, static recovery, and flow cytometry. We'll explore these later in the presentation. Based off of that experiment, the following results were collected. There are two major takeaways to be taken from the collected data. The first one being that a dynamic culture system can produce exponentially higher cell counts when compared to its static counterpart. The second takeaway being that there are no statistically relevant differences between the cell counts obtained from the seed train condition when compared to the direct thaw condition. From here, we can move on to the cell validation. Our first form of cell validation is trilineage differentiation. This refers to the cell's ability to differentiate into bone, fat, and cartilage. As you can see based on these images, the two conditions tested did not affect the cell's ability to differentiate into any of those cell types. Next, we move on to flow cytometry. Flow cytometry uses the principle of fluorescence to test for specific surface markers on a cell. The plots at the bottom of your screen represent the gating strategy that was used to collect the following information, and the bar plots above represent positive populations for each of the surface markers tested for. These were CD73, CD90, CD29, and CD44. As can be seen by these plots, both conditions produced extremely high positive populations for all positive surface markers. Based on the data presented today, I'd like to summarize the main takeaways. The first one being that MSCs are a possibly viable treatment for cartilage injury and osteoarthritis. The second one being that cells can in fact be directly thawed into stirred suspension bioreactors while maintaining their trilineage differentiation abilities, their immunophenotype, as well as their ability to proliferate. Finally, I'd like to thank all members of PPRF, Sparks Lab, as well as Crowitz Lab for assisting me on my research journey so far. I'd also like to thank all resources used within this study, as well as all the funding sources that have made my research possible. I thank you everyone for listening today and enjoy the rest of your days.